I am delighted and honored to be able to join you to speak about air pollution. In 2018, the World Health Organization made a report, and I'm going to read from it. The combined effects of ambient, outdoor, and household air pollution cause about 7 million premature deaths every year, largely as a result of increased mortality from stroke, heart disease, chronic obstructive pulmonary disease, lung cancer, and acute respiratory infections. But what is the effect of air pollution on the brain? Obviously, it's no good for our lungs and our respiratory system, but what do we know about its effect on the brain? This is a topic which has not been that much studied, but it's just becoming now increasingly studied. Let me tell you something about this. Here's what is known. In recent years, a growing number of epidemiological studies have shown that fine particulate matter, I'll be calling that PM, with aerodynamic diameter of less than about 2.5 microns, that's abbreviated PM subscript 2.5, may increase the risk of dementia, including Alzheimer's disease. Okay? Pathological studies suggest air pollution accelerates amyloid beta accumulation in mice and neurofibrillary tangles in dogs. Okay, despite the increasing evidence supporting the neurological effects of PM 2.5 on Alzheimer's disease and related dementias, definitive conclusions on the causal association cannot be made because the underlying mechanisms remain unclear. Now, this is from a study that's made, published in Brain this year, 2020. I want to show you something, of course, about the mechanisms involved and think that we are going to describe to you in advance in this area. Here's more about the results from this article. They looked at 998 women, ages 73 to 87. These women were free of dementia, and if anyone got dementia, they were removed from the study. They were studied by magnetic resonance imaging of the brain, and they were given periodic memory tests. This study took place 11 years. The scientists found that the greater the exposure of the women to PM 2.5, the lower their scores on cognitive tests. The MRI, that's magnetic resonance imaging results, showed that increased PM 2.5 exposure was associated with increased brain atrophy, even before clinical symptoms of dementia appeared. So obviously, we have a strong correlation from this that polluted air really affects one's brain in humans. It's hard to study human brains, and we actually turn to mice. This is my research group. All the work I'll be telling you about was done in my satellite laboratory at Fudan University in Shanghai. And I will be speaking particularly about Dr. Sarah Hamid, where my arrow shows right now, who carried out the majority of this work. And here's the plan of the experiment. 8 mice were exposed to ambient PM 2.5 particles 8 hours per day, 6 days per week for 24 weeks in enrichment chambers with real-time mass concentrations of this PM 2.5 around 70 micrograms per cubic meter. And the number of PM at 0.1 microns in diameter was about 10,000 to 20,000 per cubic meter. Another 8 mice breathed filtered air during the same time period. Then all mice were sacrificed and an analysis was made of mouse brain tissues, slices of the mouse brain. And I need to remind you, this was 70 micrograms per cubic millimeter. Actually, here is a plot of the Shanghai air quality from 2014 to 2019. I don't include 2020 because the coronavirus pandemic has really changed the air quality. And I want to compare with normal air quality. And you'll see it's around 35 in 2019. So we're looking at a level about twice that, which is the average value which in Shanghai still exceeds the World Health Organization annual limit. Let's see what happens. And here are the people that are involved. Shown in the middle is Professor Jin Zhao, who is an associate professor in the Department of Environmental Science in the School of Public Health. And on the right is Dr. Zare Hamid. Here is the exposure chamber. I'm just showing you what it's like, where you can see the mouse cages are kept, and here are the mice. So you see that it's very real. And now we turn to the results. Let me give you quickly some of the results. We found that PM particles from dirty air entered mouse brain, which was visualized by field emission scanning electron microscopy, SEM, scanning electron microscopy. And let's look further at this. First, the mechanism. What happens is you wonder, how do the particles get into the brain? They're supposed to be a good blood-brain barrier, but they're getting in there somehow, as we're going to show you. And it appears to be primarily coming at first through the nose of the mice during breathing. There's mucus secreted, and there's a motion of the cilia that actually push the particles into the brain. And so this is the translocation of PM particles into the brain through olfactory neurons inside the mouse nose, and the movement of PM particles in the blood capillary through brain cells. Okay, and the next picture I'm showing you, D all the way through L. D is a surface evaluation of mouse brain tissues by the scanning electron microscopy, in which brain tissue from filtered air showed no abnormality. That's D. Look how blank it is. But now E through L are brain tissue from dirty air, which showed very much on the surface that you get this. The scale bar is about 1 micron. We're really seeing damage in the brain, unquestionably. Okay, let's look more at this. This is the evaluation of the topological features of mouse brain tissues. A is the mouse brain tissue from dirty air, and it's showing inflammatory cells attaching with microvillar projections of endothelium of the blood vessel. B is a magnified image of the blood vessel in the brain. Okay, and C is the exposure of particulate matter to brain activates the immune response and enlarged perivascular space. It appears that there is a protective response, an immune response, but as this goes on, unfortunately, this immune response, this inflammation, leads to more things happening which are deleterious. Okay, and here's the next picture I want to share with you. This is the evaluation of the topological features of mouse brain tissues. And D through I are observations of anatomical features and topological alterations suggesting neuroinflammation. And I'm showing you the different scale bars, and it's pretty clear what's happening in this situation, that there's real troubles. Okay, here's now some staining experiments. Now we're going to stain with things. One of the normal things to stain with is H&E, stained mouse brain. That's in A, from filtered air, and it showed no abnormalities. That's what you see in A. Whereas B through D from dirty air showed neurofibrillary tangles and plaques. And now in E, we start to do Congo red staining of mouse brain tissue. And again, no abnormality is seen in E, but look at F and so forth. And we see mouse brain tissues from dirty air showed accumulation of inflammatory glia cells and amyloid deposits. Now we use antibody staining. We use immunochemistry with antibody staining of mouse brain with dirty air, and they showed these amyloid deposits, the type you see in the same way in Alzheimer's disease, okay? And the scale bar is 4 times magnification in A and B and 10 times and so forth. Okay, so we get a sense that we really have seen this in the brain, and we now have anatomical evidence of what's happening. People have already seen some of this before. Now, what is really new in this study is shown on this slide. And I need to explain this technique that I have been pioneering, though it started in Professor Graham Cook's lab at Purdue University. And it is called DESI-MSI. That's desorption, electrospray ionization, mass spectrometry imaging. Here's how it works. There's a tissue that's mounted on an XY translation stage. That's at the bottom. Okay? We send in a droplet. The droplet strikes the tissue. Now it makes a splash, and we catch part of that splash into a mass spectrometer that will look at the masses of all the things that the droplet dissolves. We pick the droplet to be some solvent which picks up lipids. We can give technical information that it's a one-to-one mixture of acetonitrile and dimethylformamide. Never mind. We're going to use this and get now a mass spectrum, and we're going to apply this and make an image. And we're going to make the image by, as you do with a TV screen, and you raster across. We're going to move and translate the tissue, taking pixel by pixel and making an image out of it. And here showing you in the middle is such an image. At the top, we see the mass spectrum. And let's look some more at this. This is profiling of metabolic alterations in mouse brain. Mass spectra of brain samples from the exposure groups. A is dirty air. That's the red spectrum. And the bottom part of it in black is the filtered air. There are some clear differences. B and C are ion images of what's called ceramide and sulfatide, okay? And there's a color scale that in which pink's the most intense and black is no signal, okay? That's trying to show, if you look at a mouse brain where it's been distributed, you'll see it's a lot of it's all over. And now we're making an F. Take a look at panel F. This is a quantitative signal intensities reported as the mean plus or minus 1 standard deviation. And we're looking at ceramide and we're looking at sulfatide of these two particular types. And we're seeing that they're greatly upregulated. That's the black for the dirty air compared to the blue, which is the filtered air. Okay, we're really seeing this. We're getting now chemical information about a change that's taken place in the brain caused by exposure to these fine particulate matter. And this is showing yet in more detail for more compounds. Okay, and we can see all that's happening. I can go through this in more detail, but it's important to know what are these compounds, what are they like? And for some people, this will mean a lot. And for others, I just want you to know that we know. And that's what's shown in the next slide. These are the compounds, for example, that we're seeing. Where CER is the ceramide and the STs are the sulfatide type compounds. They're different and we see them as I've shown you. And they're all upregulated. Okay? Now, what does it mean? What's the significance of ceramide upregulation? Here's what we know. Ceramides play an important role in killing cells, apoptosis, differentiation, and inflammation. Previous studies have detected upregulation of ceramide molecules in injured rat brain. Moreover, the studies showed that ceramide and brain damage co-localize. So do we find that, suggesting direct role of this sphingomyelin metabolism in neuronal cell death. Ceramide is the main product of this sphingolipid metabolism, and it is directly involved in the aggregation of amyloid beta, okay, for the progression of Alzheimer's disease through stabilization of the enzyme beta secretase. As a result of positive feedback mechanism, the generated amyloid beta activates the sphingomyelinase enzyme that catabolizes sphingomycin and increases the ceramide levels in the brain. So it's unfortunately a positive feedback mechanism. And ceramide also activates a number of biochemical processes leading to malfunctioning of mitochondria and generation of ROS, reactive oxygen species that stress the system. Okay, so that's this ceramide. Now let's look at the other. What about this thing called sulfatide upregulation? That's actually sulfated a sugar, galactosyl ceramide, also known as sulfatid, are found in abundance at the myelin sheath of brain cells. We found upregulation of this material in the brain tissues from dirty air compared to the filtered air, suggesting damages in the myelin sheath and blood-brain function dysfunction as a result of particulate matter exposure. This hypothesis is supported by previous reports that have shown that the expression levels of sulfatids are associated with the physiological activity of the blood-brain barrier, and increased concentrations of sulfatid has impact on myelin sheath at early stages, for example, in the HIV-1 infection. So this is also no good, okay, that we're finding. And we're understanding that now, even though this may start by coming from the nose, it's going to open up the blood-brain barrier and allow more to come in, which is evidently what we're seeing, I think. Okay? And so now let me reach some conclusions for you, if I might. Here they are. Inhaling air containing particulate matter, PM 2.5, is known to be deleterious to health, particularly to lung function. But this study presents, I think, striking visual evidence of its harmful effects on brain tissue. Our study shows systemic inflammation in response to accumulation of PM particles in the mouse brain, resulting in amyloid deposits and the formation of tangles and plaques. We use this technique of desorption electrospray ionization, mass spectrometry imaging, DESI-MSI, and it revealed upregulation of the ceramide and sulfatide, both associated with brain damage. Our findings strongly support the contention that prolonged exposure to PM 2.5 airborne pollutants increases the risks of cognitive decline and dementia. And so I reached finally the following conclusion. I want to raise the alarm about this menace, which is pervasive in urban settings. We all think that air pollution is bad. I did not know in this study until I got deeply into it, that it's not just a matter of affecting your lungs, it really can affect your mind. And I think that's a very serious matter. One that people who set public policy need to pay attention to. Thank you very much for giving me this opportunity to present to you this bit of work that we've been doing, all done at Fudan University in Shanghai.